안녕하세요. It's very nice to be back in Korea. Probably, if not my most favorite country. I spent quite a few years here as a postdoc, so it's always a pleasure to come back. Yeah, thank you all for attending today. In my session, we're going to, if I can figure out how this, oh, there we go. I'm going to speak to you about the Cellscape XR and essentially the spatial proteomics platform that we have at Bruker. So my team develops this system. It's called the Cellscape XR. It's what you see here over on the far left side, on your right-hand side of the screen. And this is a new platform that we launched this year. And so that's the theme of what we'll be talking about here. Okay, so that's how this works. I'm sorry about that. So let me start my talk by showing you this slide. And I'm going to make a statement, and that is that multiplex immunofluorescence is the benchmark for spatial biology. I think it is true today that if you do a spatial biology experiment, especially a spatial transcriptomic experiment or an ROI-based NGS type of spatial readout experiment, you're going to have to validate your data downstream. You will need to convince your colleagues in pathology or in the clinical department that your findings are indeed real, that your cell segmentations are accurate, and that your cells actually exist. And this is a proof right here. If you look at these papers here, and these are all from like January or February of this year, all of these papers used some type of really exciting spatial biology technology, transcriptomics, proteomics, you name it. And they made some nice findings. But they all have one thing in common. They use multiplex immunofluorescence, or IHC for that matter, to validate their findings, most generally in the last figure of the paper. So if you want to publish in Nature, if you want to get your stuff out there past peer review, you need to validate your data. And most, if not all, researchers eventually turn to immunofluorescence, or MIF, multiplex immunofluorescence, which I now consider the benchmark for this. Why is that? It has extremely high fidelity. It's very robust, it's very repeatable, and it's a quantitative extension of IHC. Hence, it's understandable to a pathologist. The data format is the same. We're counting photons, we're generating photographs. It's the same as what IHC or pathologist sees. So it's not a rendered result. So they do not need to interpret the data. They can look at them, they can see it. They can see the result in order to believe it. Now, in our world, we would want anybody to be able to do these experiments easily, but they're not always that easy to accomplish. To this day, there have been problems with throughput. Some of these assays have been slow. Some of the assays have been harsh. They've been damaging the tissue. Customization has been limited. Customization is really, really important because everyone in here is going to study a different topic. You're going to look at different biomarkers, so you need to be able to customize your assay. And this has really been a pain for a lot of researchers. And those are the questions that keep myself and my team awake at night. And those are the questions that animated our launch or our development of the Cellscape XR platform. This is a completely new spatial proteomics platform that we announced in February of this year. There are a lot of really, really cool features that are packed into this box. And I can't go through all of them with you today because I simply do not have enough time. But there are going to be three features or three aspects in this talk that I'm going to be speaking to you about. One is this is a quantitative image system. So you're not just going to be looking if a biomarker is there, you are able to measure how much of it is there. That is extremely important, especially, once again, if you're working with colleagues in pathology. The other one is, and I'm going to show this to you and hope to convince you that we have an extremely robust assay chemistry, yet one that is very flexible and agile. One that allows you to build an assay with as much customization as you need. Not kits. Kits are great. They work, they're validated, they're fast, but they're not for everyone. Everyone needs to customize someday, and I hope I can convince you that you can do this on this platform. Last but not least, I would like to say that this system is built with clinic in mind. Someday, we at Bruker believe that these systems will be used to diagnose diseases and disorders. So we need a system that can go to the clinic. This is probably going to be the one. So we are building this system to have good throughput, which means that we want you to do experiments in a day, maximum two, not in a week or two. You need to have good throughput. So I'll show you some of those data for all these things. So let's speak about quantitative spatial proteomics. I'm going to start with a study that was done by a clinician, Parambir Dulai, in IBD, which is inflammatory bowel disease. I'm going to use this image here to show you what these data look like. Those of you who already do multiplex immunofluorescence are probably familiar with this. I'm going to recognize some of the stuff that I'll show you. It all starts with a pretty picture. This is a pretty picture. Tell me what you want. I think we will all agree on that. However, it's so much more than a pretty picture. It's not just that. This pretty picture, just like any other type of spatial data, can be broken down into individual cells by cell segmentation. On this platform, you have a pixel resolution of 182 nanometers per pixel, which is the best on market right now. This will give you the most accurate cell segmentation. You're no longer assuming a cell is there. You're no longer assuming where a membrane is. You're tracing that membrane very, very precisely where it actually is. So it's unambiguous. Second, and very importantly, you see here, we acquire all of our data at high dynamic range. What this means is that we take multiple exposure in every imaging frames. In doing so, we extend the capture range of our camera. And if you have a biomarker signal that is, for example, PANCK, that's very, very bright, that exceeds the capture range of a detector, you need to take multiple exposures. And that's what high dynamic range is. When you have high dynamic range, you can capture biomarker signals that are high, low, medium. Essentially, you can quantify your data. This is unique to the system. This is a feature that we're very happy about. This system is very good throughput. It works with standard histopathology slides, any superfrost slide or whatever you have in your lab, which means that if you put a lot of tissues or large TMAs on here, you're literally studying millions of cells in a single experiment. So that's pretty good. Last but not least, we call this quantitative spatial phenotyping, which is the culmination of these data features. They give you probably some of the nicest representative, highest quality spatial proteomics data that are available anywhere. So let's have a look and see what this looks like based on Parambir Dulai's study. As I said, he studies inflammatory bowel disorder, and Joe spoke about some of this work already. So what he does is he uses actually kits from us. You can see these two circles up here. Those are our antibody kits that are readily available to do both immune and architecture profiling in his bowel disorders. And once again, it all starts with a beautiful picture. And in this beautiful picture, you can see in cyan color, CD45RA, immediately it pops out as an immune infiltrate. So what we do, just like most others that take this type of analysis, we segment the cells, we cluster them, and we do a spatial neighborhood analysis. Here you can see 5 neighborhoods, MB, 1, 3, 4, 5, and sorted. You can see that immune infiltrate. Pay attention to that. It pops out right away, NB5. You can see some immune cells in here. It's pretty much what you'd expect from this type of analysis. It's very nice. So obviously the experiment works. But here's where the cellscape differs. It's not just about saying that a biomarker or a cell type is there. It's about measuring biomarkers within that cell type and then making assertions. And I'm going to give you this example based on Tregs. You can see on this, on the left-hand side, let's pay attention to the bottom left image. This shows you 3 Tregs. The one on the bottom left, very bright FOXP3 expression. That's a Treg with FOXP3 high. Then you have these other 2 Tregs. Those are FOXP3 medium or low. Now, if you take all of those, several thousands of these cells, and you plot CD4 on the x-axis over on the right versus FOXP3, you can now see a distribution of FOXP3 signals, different Treg states. It's no longer just a question whether or not we're seeing Tregs or if it's just one cell type. We're actually now watching a functional progression of this regulatory state. And if you've ever used a flow cytometer, you'll recognize that you can actually draw gates or circles around these different levels of expression and further characterize your cell types. So we're not just looking at a phenotype now, we're looking at a functional state. And this is done just with MIF. We're not even imaging the transcriptome right now. That's really, really important and really, really powerful. So with that in hand, and Parambir Dulai, coming back to this neighborhood map, which is in some sense canonical, this is what we know, he went back and said, you know what? What if I actually reanalyze this tissue and look at the Treg state? It's a completely different immune profile. It is no longer about one large immune infiltrate that you see on the left here in cyan. Now you have a representation of potentially pro-inflammatory and inflammatory processes across this entire tissue. And if you look at these maps down below, and you look at different types of immune cells, for example, on the right-hand side, the 5th biomarker on the bottom, CD56, NK cells. There's not very many NK cells in this tissue. You can see, however, if you look at a FOXP3 negative neighborhoods, this is essentially CD4 T cells that are not Treg state, there's CD56. If you look over on the other map, where all this information is combined together, CD56 doesn't even show up because that information has been diluted. So this is both more precise and more informative all in one shot. Once again, this is solely based on immunofluorescence imaging, and that is extremely powerful. Now, I should add here that this isn't just immunofluorescence imaging, and I'm going to talk about this a little bit more. But what Parambeer did upon discovering these patterns is that he took his cellscape image or his cellscape data. So first he did the cellscape experiment, about a 25 antibody panel, and then he was interested in these different types of Tregs, and he asked, can I take this same tissue and now put it on the cosmics? And that's exactly what he did. He took the very same slide after cellscape, put it on the cosmics, and ran a 1K assay. And if you look at the bottom right histogram, he's actually now able to look at the transcriptomic profile of these different Tregs, cell by cell. He can see the Treg on the cellscape data, and he can see it on the cosmics and correlate certain types of inflammatory markers and pro-inflammatory markers to them. And as we know today, these states actually check out. So that the different levels of FOXP3 expression that we catch in immunofluorescence are indicative of functional states. So this shows you, A, quantitative imaging is really informative and potentially very, very important or redefining of what we do in this. B, that this assay is actually compatible with downstream cosmics and experiments. And I'll speak about that a little bit more. So this is the first sort of data that I wanted to give you, and that's really, this is discovery research. I mean, you discovered something new right here. This is really cool, and this is going to go on. There's a large-scale cohort study planned for this now that I'll get into now. Let me show you a couple other use cases that are now probably many researchers are using the cellscape downstream as a validation tool. So let's talk about that a little bit. So as Joe already talked to you about earlier, researchers like Miranda Orr that used a geomics system, which is really a fantastic high-plex system. It's got great throughput, it's robust. I mean, that's a super discovery tool. She's using that in Alzheimer's and other neurodegenerative diseases to find biomarkers that are expressed in brain tissues. And then once she finds biomarkers, like over there in the top right, you can see about 30 biomarkers on a heat map. She wants to go in and look with single cell resolution what cell types these biomarkers are expressed at. And in immunofluorescence, this is unambiguous. You're imaging an entire brain slice. You're looking at millions of cells. You can see whether or not a biomarker is expressed in a neuron or an interneuron or a glia. It's exactly what these slides show you here. And we are able to translate panels from the geomics system onto the cellscape system, sometimes in a matter of days, using the same clones. But generally, for a 20 to 30 plex assay, we need about a month to build a panel. This is still, by any stretch, fastest panels that you will ever build. So that's useful. And you can customize it. You can add or subtract markers as you need to. But this picture, and there's a paper here in which they describe this type of validation, this picture will convince the reviewer that whatever you see in your heat map is actually expressed in the brain at the single cell level. Nowadays, you need that. You can't just publish heat maps anymore. You need to have single cell validation. And this is the best way to do that. Here's another study that's been uploaded to bioarchives, came out of the National University of Singapore. So Anand and his team, they study a central nervous system lymphoma. This is a tumor that has really strict tropism in the brain and has virtually no treatment options. And we don't know a lot about it. What Anand did is he started his study with the whole transcriptome assay and the geomics to characterize 18,000 targets in these lymphomas. And what they found is that there's a lot of macrophages in these tumors, not as many T cells, or there's no difference in T cells in comparison to other lymphomas. So there's something going on with the myeloid cells and the macrophages. The bottom graph here shows you a pathway analysis that they did on the whole transcriptome analysis. And this pathway analysis indicated that there was a certain type of immunosuppressive macrophage in these brain tissues. And we call this a lipid-laden macrophage, or it's a foamy macrophage or fatty macrophage. There's some acronyms for that. But it's an interesting type of macrophage that's not commonly, we don't commonly stain for them. They're different. I mean, there's all kinds of different immune cells, but this is, let's call it, a new immune cell to us because we don't have antibodies for it. So Anand has to convince the reviewer that this macrophage really exists in his sample. You cannot just have a pathway analysis like this and conclude your paper's not done yet. Now you need to validate it. So what he did, took his cohort, used a cellscape system, and first confirmed that there's quite a few macrophages in that. That will be the second histological image here. You can see CD163, CD68, of course, with a lot of B cells in there as well. Indeed, there are a lot of macrophages in this tissues. But it's just asking whether or not they're there. Now what Anand needed to do, he needed to confirm that there's a LLM, a lipid-laden macrophage present. And in order to do that, you need to stain the tissue with markers like GPNMB, TREM2, and other markers. These markers are not currently present in our kits. So what Anand needed to do, he needed to buy these markers and just add them to our assay. And this is one of the really cool things about the cellscape. You can buy your own antibody and you can plug it into your assay in order to find the cell type that you need to find. You don't need to wait for us to give you the antibody. You can just go to Abcam, CST, Thermo, wherever you want, which is exactly what he did, and just incrementally added more of these antibodies into his assay until he found, you can see here, LLM cells versus non-LLM cells. And nicely enough, these cells have some type of a spatial signature that correlates with patient outcomes. This is exactly what researchers want to accomplish. So this is one example of someone who's essentially customized his way to an answer. Very important and something we support. Here, one more paper just to show you what this is used for. This is Rong Fang. Excuse me. Rong Fang does a lot of spatial transcriptomics imaging. So his is ROI and NGS based. And what they did with their transcriptomic technique, they found a B-cell lymphoma process that's called a Richter's transformation. And what happens is in a Richter's transformation is that a chronic leukocyte lymphoma, which essentially there's something wrong with the B cells there, but it's non-fatal. Something happens to those B cells and they turn into diffuse large B cells, which is a fatal disease. People die and we don't exactly know how to treat it. The in-between phase he characterized using transcriptomics. But in order to confirm that these are the right cell types, this is really happening, he needed pathology-grade immunofluorescence data. And that's what you can see here on the top right. On the left, you can see the cellscape data, very precise morphometrics and imagery, really, showing you what the chronic leukocyte lymphoma looks like. And then you switch over to the right and you can see these big, ugly, nasty B cells. That's what DLBCL looks like. This is a pathological definition of it. This figure was added to the paper and got this in the Nature Methods. Without this validation, he could not confirm that he's looking at this thing. One more thing to add about this experiment is same section multiomics. So he first did spatial transcriptomics on his dbit-seq, used this same section, stained it with multiplex immunofluorescence, and thereby was able to do single-cell spatial transcriptomics. So that's a really nice study here. I encourage you to have a look at that. So let's talk about the assay. I think this is really, like Joe said, the most important decision that you make about any type of spatial research is think about your assay. Get an assay that's agile, get an assay that's robust, get an assay that you can trust. And especially buy a box that's built around an assay and not the other way around. So the CellScape XR is built around our new EPIC IF assay. So let me talk to you about how this works. Most of our antibody panels, and there's different ways to build multiplex immunofluorescence panels, but most of our panels are built around direct immunofluorescence. And this is by design. There are thousands of antibodies available that have fluorophores already conjugated to them. You can buy them yourself from antibody vendors. You can conjugate fluorophores if you want. We sell kits, so there's a lot of material available. We can probably answer any question in this room with already existing content. It's highly specific. There's no cross-reactivity, no host-host interactions, none of that. There's no stipping. There's no extra steps needed. And if you have the right photo bleaching methods, it means that you can actually do really cool stuff with this. And photo bleaching is the key here. For many years, people have tried to use photo bleaching to remove fluorophore signals, and it was not always very effective. About 2 years ago, we launched what's called Epic IF. Epic IF is a completely new way to do photobleaching that has not existed until we published that. So this is a patented now way to use expansive photobleaching. What this means is that any organic fluorophore that people use nowadays for immunofluorescence or IC or fish or TSA-based assays, any organic fluorophores in any wavelength are compatible with this system, meaning that we can remove Alexa Flow 48, Alexa 750, 647, 555, you name it. It doesn't just have to be Alexa. It can be Dialite, Coralite, Addo, whatever you have out there, whatever you buy is compatible with this system. It's reliable, meaning that within a few seconds, we get complete erasure of the signal. It's robust and it's very flexible. You can use it on different types of assay, and I'll show you some of that data. But the combination of direct immunofluorescence with this is an incredibly powerful way to build an assay. So this is what this looks like when you put everything together. This is your sample preparation. So this is the whole platform put together now. The experiment starts with your sample preparation. You're responsible for dewaxing, baking, antigen retrieval. Thereafter, the sample goes onto the system and is fully automated. We stain it. The CellScape acts just as much of a stainer as it is a microscope, so it does the staining for you. It delivers the antibodies. These, of course, have a fluorophore on it. They label your cell types. You take a picture, as is shown with the camera, and then the photo bleaching happens, as is shown with the light bulb. So this, I'll show you that. You can do about 4 or 5 colors at a time. You could try 7 if you want, but that's a little bit audacious. But that's all there is to it. This is a very simple assay. There's no tricks involved. It's very transparent. And the Epic photo bleaching, of course, is the key thing. And this is unique to our platform. Other platforms use stripping or barcoding and other ways to remove signals. This is the only platform that is 100% committed to photo bleaching right now. So this is now what everything looks like in the real world. And let's walk you through this. So on the left, you can see the CellScape. There's a microscope, and then that other box next to it is the fluidics box. So this is where the antibodies go and all the buffers go, and it's all delivered. The picture above, it shows you what the CellScape looks like when the hood is open. And you can see there's a stage in there. On that stage, you can fit, oops, sorry. I just pushed the wrong button here. This is a strange thing. Here we go. This laser pointer isn't working. But anyway, you can put 4 slides into the system in parallel, and they can run in parallel. That increases your throughput. If you look at the bottom here, these are the excitation emission spectra for the channels that we have. So there are 7 channels in here, going from 425 to 750 nanometers. That covers just about any excitation emission range that is commercially available today. You can use these channels at will, so it's up to you how you occupy them, what types of antibodies or fluorophores you put into your assay. And now you can see in the middle here, DNA, CD3, PanCK, and so on. This is what a typical cycle looks like in a CellScape experiment in my lab. They put 4 antibodies in there plus a DNA marker. So 4 or 5 antibodies plus DNA makers, very standard. And that's one cycle. You do another cycle, you double it, and then you triple it, and then you can rise your plex up. I'll show you that. But pay attention to the image on the orange image here on the top. Once we take the picture, once you have all this data recorded, now we do the photo bleaching. And now look at the orange. This is real time. This is very fast. It's about 2 seconds. This entire signal from the nuclear markers has been removed. All that's left are little red spots, and those are red blood cells. That's autofluorescence. That's taken out with an image subtraction later. The system is not designed to remove autofluorescence. But that fast photo bleaching means that you have very, very good throughput, and it means you can cycle your system quite efficiently. So that's quite nice. Building on this, we have a variety of antibody kits. They come in this format. So the antibodies are all screened, validated, and very heavily validated. I think they sell the kits for about 10 reactions. So one kit allows you to do 10 experiments, and it's been tested. And we test this on dozens of different samples. We provide information on dilutions, suggested what you need to do. So the optimization, always need to optimize, obviously, but it's relatively minor. And this is a fast-growing selection of kits. Most of the work that our customers do to this day is immune profiling and cancer research, but we're also building kits for neuroscience now. One of the other things that we launched this year in February is called VistaFlex. Now, VistaFlex, I think, is a very, very powerful addition to this portfolio. What VistaFlex allows you to do, and this is an immunological label. So let's assume you have an antibody in your institution or some type of an antibody that is not available in a preconjugated format. It happens all the time. People still need a way to label those antibody, and this is what VistaFlex does. This is quite nice. So what you do is you take this, and we provide this as a kit. You take one reaction worth volume of your antibody, label it with VistaFlex the day of the experiment. It takes about 10 minutes. And it's one reaction worth. You don't need 100 micrograms. You don't need 1 milligram. You can start with as little as 1 to 2 micrograms, which is amazing because that means it's cheap and it's fast. And then you can plug and play it into your assay. It's immediately compatible with existing VistaPlex kits. It's probably compatible with every custom kit you have. So this is a nice way. Any antibody you have, as long as it is an IgG, can be deployed on the CellScape. And just need to label it with your kit. Takes 10 minutes and does not take a chemistry degree. And that's quite exciting. And we work with this kit. So this is an example that shows you here on the left. This is a squamous carcinoma of the skin. And this is what it looks like when you label it with existing CellScape antibody kits, essentially immune cell lineages in the architecture. But the investigator that we worked here with, they were interested in cell metabolism. And there were 6 antibodies that they wanted to add, including GLS, LDHA, PDK1. And essentially, we didn't have those antibodies. So what we did is we labeled each one of those 6 antibodies with this VistaFlex technology, and they just plugged them into the assay and ran the experiment. And you can see, if you look at these 2 images, these big ones, this epithelium here, nice stratification with the general kit. It's sort of yellow and turquoise and green. But if you add these metabolic markers, the amount of information that you get is just so much denser. And this is really, really cool that you can do this. Now, let me emphasize, this is a serial experiment. And let's talk about this a little bit more. This is one of the really, really cool features about the CellScape. Remember, it's a cyclic immunofluorescent. Every cycle you can regard as an independent experiment if you want to. And this is how that works. What you see on the top here, this is an image of a brain slice, human FFPE. And what we're doing is we're building an antibody panel for someone. And this antibody, N-neun, N-E-U-N, is a monoclonal antibody we want. It should be pink, and it should be in the nucleus of the cell. This antibody did not stain the nucleus of the neurons in the first cycle. So essentially what happened is we ran the CellScape, and then we looked at this picture, and we concluded that this antibody is not working. And you can see that golden epitope here. This is the structure of the target molecule. The golden epitope is what this antibody is targeting. But it doesn't work. But it's no problem. What happens is, if you do this experiment, you can take this tissue and you can park it in the refrigerator, and you can do exactly this analysis. You can talk to your colleagues, or you can forget about it and start it again someday. It's up to you what you want to do. So we did this, and when we recognized that we can buy a different antibody, you can see in red for another epitope, we took this sample back out of the fridge and restained it. And then we put it out of the fridge one more time and restained it with a polyclonal marker. This is perfect staining right here, Ola. But think about it now. You're building an antibody assay. Normally what happens if you have a negative result, you have to throw the slide in the trash and you start again. And then you don't know if it was because it's 2 different tissues or something. But on a CellScape, you can stop and go, cycle by cycle. You can start your experiment just using 4 antibodies. And I would always recommend that because you don't know how good your tissue is. And if you're happy with the tissue, you can go on and on and on and on as much as you want. And Nick West, he's going to give a talk later today, I believe. It's exactly how we worked with him. He came to us with an unknown tissue type. These nasal polyps. We've never worked with those before, so we took a cautious approach. We first ran a few cycles, showed that there's a beautiful immune profiling, beautiful immune cells in there. It's a really cool study. But Nick then was interested in what's called an eosinophil. A, we didn't have antibodies for this. B, we didn't know Nick nor his tissues. So we took this stepwise approach. And essentially between the first, second, and third picture, approximately 2.5 months passed with increasing addition of antibodies. Some that we already had in the fridge, some that he bought for us. So like CD15 is an eosinophil mark. You can see this beautiful eosinophil here in purple. And CD66, those came from Nick. This is what he needed to look at his stuff. But it's just like you're building a puzzle. And that someday that puzzle is complete, but it is only complete when the biology tells you to stop. It doesn't stop with, bad tissue or bad panels. And this is incredibly powerful, especially if you're using, if you're in a core lab and you're working with a lot of different tissues or unknown stuff. So that, I think, is a unique feature about the cellscape, and it's definitely worth talking about. And I think Nick will talk about this biological significance of this a little later today. One more thing, I hope to convince you that this is a quite agile system. So you can't just do immunofluorescence on a cellscape. You can also do same-section FISH. So same-section multiomics using immunofluorescence with up to 12 FISH probes. And this is an example of that. This is an experiment that we're doing with St. Jude. And essentially what we're doing here is CAR T cell profiling in a mouse xenograft model. So the idea here is that we use a cellscape just like we would to do our immunofluorescence experiment, but in addition, we're labeling FISH. And why are we doing this? Because CAR T or CAR receptor sequences, they're unique. Whichever investigator institution you work with, these are often proprietary, and it's cheaper and faster to raise a FISH probe to them than to build an antibody. So the question was from Jasmine's team, can you do FISH and protein imaging on the same sample? And the answer is absolutely yes, and it's actually easy to do, and this protocol is published. So here you can see the xenograph. This is a tumor sitting in a mouse skin. You see the tumor in white with the margin. You see lots of CD8 cells hanging around out there, some of them infiltrating CD4, and lots of granzyme B. So there's definitely a kill signal in this tumor. Pretty cool. Now in red, you can see the FISH signal, same section, up to 12 probes. That's where your CAR T cells are. Now you combine that with really beautiful high resolution imaging. You can see on the bottom here where the white arrowhead is. Turns out that these CAR T cells are actually CD4 T cells, and we expected them to be CD8. They're not CD8 at all. They are CD4 T cells that express granzyme B. So here you can do your spatial neighborhood analysis. You do quantitative imaging, whatever you want. When it is same-section multiomics, fully customized. It's up to you what you want to do. You can raise your own probes. You can buy them from someone's catalog, up to 12 probes, plus however many antibodies you want to add. So let's talk about that. How many antibodies can you add? How robust or how gentle is this assay? We spend a lot of time evaluating tissue integrity on our system because the obvious concern that people have is does photobleaching damage to tissue? The valid concern, heat should damage tissue. But remember, we're only photobleaching for seconds. Like it's so fast. So the amount of thermal energy that we're delivering onto the tissue is quite minimal. A pathologist agrees. They evaluate these HE stains routinely for us, and so far they have not found any systemic damage. I think a more interesting result is what we had presented this year at AACR. And this poster, we showed a 208-plex study. So that's essentially 200 antibodies delivered onto the same TMA. This is the highest multiplex immunofluorescence experiment that has ever been done to date. 2 years ago, we published a 102-plex study, on a different platform, a different group that I was part of. This is double that, and this experiment is not yet finished. I think as of yesterday, they were up to 237 or something. I don't exactly know the number, but this continues to grow. You could not, if you were systematically damaging the tissue, do this many antibodies and get good signals. And really, the point here is not to do high plex with the cellscape. That's not the main application. It's just to demonstrate that we're not damaging our tissues. The other thing I should mention here is that 100% of the antibodies that we used for this assay were sourced from a third party. It's all Abcam, CST, thermal, whatnot. Straight out of the box. With little optimization, we built a 208-plex panel in about 3 months' time. It's fast, it's reliable, and it's never been done before. So this is quite nice. I think it shows you both flexibility, robustness, and speed, all of these features that we really wanted to accomplish with this system. We already talked about this, so we can build a high-plex experiment. You're not damaging your tissue. Obviously, this enables people like Parambir, which I started with, to take cellscape slides and put them downstream on the cosmics. This is just beginning. Everyone's always saying, oh, can I do immunofluorescence after, cosmics or xenium or this and that? I urge everyone to think the other way around. Protein is really where it's at. Protein is really, really informative signal. There's so much information content within protein. Now we understand how to analyze a whole transcriptome, but we're just now starting to reach the frontier in protein. I think this is actually infinitely more interesting, but I'm biased. I'm a protein person. But for those of you who are familiar with the geomics, I think this is an interesting application here. So what Miranda's team is doing today, they have two cellscapes in their lab. And so what they're doing is they're imaging on the cellscape. It's about 25 antibodies. And then they're taking those same samples and putting them on the geomics now. And why does this matter? If you pay attention to the yellow image on the bottom, that's the geomics image of a neuron. So in the geomics, you have 3 structural markers that allows you to see neurons, see plaques, and other type of things. That's pretty good, but the information content is limited. Because if you look at the top, just go straight up, you can see that same neuron in cyan. But you can also see that yellow cell that looks like an octopus that's sitting right on top of the neuron. That's a microglia. That microglia is sitting on top of the soma of the neuron. This means this neuron is not normal by any means because there's a cell sitting on top of it. So if you draw your geomics ROI on there, you're actually looking at 2 cells. It turns out that the data that comes out of that is completely different for this type of neuron versus another neuron that has a microglia. And so this is really, really cool. Now you're taking your discovery research and you're already subclassing it into different types of discoveries. It would not be possible if you weren't able to marry these 2 platforms. So if you're a geomics user, you almost have to do that from now on because this is infinitely more accurate. So that's my introduction to the cellscape. I think I have 25. Oh, I got a little bit more time here. So I think I convinced you that it's useful to quantify your signals rather than just to say a cell is there. It's useful to see how much of a biomarker is there. I showed you what I think is a very nice assay chemistry. It's robust and it's flexible and allows you to do a lot of cool things. Now let's talk about throughput in the last part of this talk. I think this is very, very important for research in general. An experiment should never take days and days and days of time. It should be quick and it should be efficient. So we've made a lot of progress in the throughput of this system compared to prior generations. So this is much faster than previous generations of ours. And I would say it has parity with any other system out there right now. It's probably among the fastest. And I'll give you this example, 2 examples on what this looks like. So Parambear's study, this is what we talked about earlier. He's obviously excited about this FOXP3 quantitative imaging now. He has decided to run this study on 200 samples. Now, this is not insignificant by any means. 200 samples is a lot. It's a huge study. So I'm going to give you exactly the breakdown for this. So the average sample size is 4.25 millimeters. Square millimeters is kind of small. He uses a 20-plex antibody panel, and he puts about 4 to 5 of these samples on one slide. And you see that on the right. That's what his slides look like. So there's 4 or 5 of these, differently sized, but yet kind of small samples on there. He doesn't spend any more time developing his assay because he's just buying our kits. Like whatever we have in the kits is good enough for what he's doing. And this is how long this is going to take him to do 200 samples. You can see on the left, we have a standard imaging mode. And if you look at the light blue bar, that's 5 cycles. That's how much he needs to get to his 20-plex. It takes him just under 9 hours to complete one of these, so about 5 samples. To complete the entire experiment, 200 samples, takes 14.5 days. So just over 2 weeks. That's not a lot of time at all. It's going to take months to analyze those data. This 200 samples in 2 weeks, there's no way that you can do this as fast in other places. Remember that, because translational or clinical research is not on N of 1 or N of 2 or 3, it's N of hundreds. And this is really, really, really important. Now, I should add that the rate limiting step for speed is the amount of area that you need to image. And so it's important to consider this. Now look at this. This is Nicholas Bless, and he's actually a clinical pathologist who's trying to develop LETs on the cellscape. So the path to the clinic has already been breached. He's already walking on it. His experiment is a little crazier. So he has 1,040 patient samples across 100 tissue types. And you can see in the middle here, this is how it breaks down. There are 520 cores on each one of these slides, 1 millimeter cores. He uses a 6 to 8 plex. That means he runs 2 cycles. So essentially what he does, he runs these by the dozen. His assay took him about a month to develop, and here you can see the instrument runtime. So imaging this in standard mode, that's at 182 nanometers per pixel plus HDR, top resolution, takes him about 36.9 hours to get the 1,040 samples imaged. That's as fast as that goes. Now, here's where I think it's important to mention the fast imaging mode. The fast imaging mode, he goes from imaging 737 hours to 13 hours. So he's more than twice as fast. This is really, really important because now he can, in 13 hours, image more than 1,000 samples. And what does this look like? This is what it looks like. On the left, you have the standard cellscape scanning mode. It's the highest resolution for one of these systems available today on the market. Top image quality. If you want best-in-class segmentation output, you need to give this as a data input. You should train your models on this. If you want to go fast and you're just looking for T cells or looking to do spatial neighborhoods in large, you can use the fast, what we call the Falcon fast mode. This image is reminiscent to what you would see on a slide scanner elsewhere. So I think that's the last feature that I want to mention is on the cellscape, it's just a single hardware configuration. You can actually switch the objectives back and forth depending on what your need is. If you need to go slow and you need to image in a lot of detail, you're going to do it on the left. You're going to be a bit slower, but you're going to be so much more accurate. But if your hypothesis is already generated, all you do is need replications, you're going to switch over to that. And you're going to do that 200 sample experiment that I talked to you about earlier that takes 2 weeks. It'll be done in a couple of days. So the throughput is there. The data quality is there. The customizability is there. If multiplex immunofluorescence is the benchmark for spatial biology, then I'm going to go one step further and I'm going to say that the cellscape is the benchmark for multiplex immunofluorescence. It has all the features that we need to really make meaningful progress in spatial biology, not just to discover, but to translate and eventually help patients. So thank you for your attention. That's all I got.